Hi everyone. Today our discussion topic is nothing but cultivation of microorganisms. It's a vast topic. From basic to the advanced, we have to concentrate. Then only it is possible to pick up the single identified colony coming to the category of the updated industrial important microorganism. So the first step is nothing but cultivation of the microorganism from the sources. We need to identify the suitable sources. From there we pick up the suitable microorganism and it will go for the several steps. First, agar medium. So in the agar medium there are so many contents are there. We need to pick up the suitable medium for the suitable bacterial growth. So for example, microbes, under the microbes virus they have a separate medium, bacteria they have a separate medium, and the bacteria also there are so many classified bacteria comes under the category of the thermotolerant, barophiles, anaerobes, aerobes. Likewise, the media, there are so many classified media. They are basal media, it's a common for all microbes. Next, differential media, selective media, and enriched media. So, based on this category, we pick up the microbes and we put in the suitable classified medium, and it will go for the further biochemical characterization of the studies. So, what are the biochemical characterization? It is nothing but to identify. Biochemical test identification is nothing but to identify the bacteria what they are producing. It might be under the category of the primary metabolites and secondary metabolites. It might be the antibiotics, amino acids, vitamins, etc. So once we identify the primary metabolites, secondary metabolites producing capability of the microorganism, then it will go for the higher level screening in the microbiology category. That is nothing but a genetic engineering part. They identify the particular gene sequence which is responsible for the production of the primary and secondary metabolites, or else in the industry important comes under the category of the industry important microbes. So they identify the gene sequence and they artificially try to improve the production of the particular product, or indirectly they try to improve the particular gene sequence expression in order to get the rich amount of the antibiotics and minerals, vitamins and amino acids. Then it will go for primary culture and subculturing, and after primary and subculturing identification, then they will identify the productability, whether it comes for the long-term usage or not. So if there is any fluctuation on the production or industry important category side, then they will go for the further improvement study in the terms of strain improvement category. So here we need to cross the basic medium construction, isolation, next primary metabolites and secondary metabolites category, next whether they comes under the category of the thermotolerant, that is nothing but extremophiles, and finally it will comes under the category of other industry used, or whether it go for the long-term stability. So aspects comes under the category of the industry viable microorganisms. Here our students they are going to discuss about the same concept in the design thinking manner. I hope that this discussion in microbiology will deliver another part of the content which is suitable for the lab as well as the high level industry.
Good morning everyone. I will talk about the empathics. First, understand the microbes, target microbes, whether the microbes is fungi, bacteria, algae, or something. And the second step is to research on the target microbe, whether fungi. The researchers are antibiotic production and probiotic environments and everything. And the third step is to obtain the biosafety level of these microbes. The biosafety level is to 1 to 4, BSL 1 to 4. The fourth and the last step is to identify the cost, time and scale of these experiments. The scale is some small scale to large scale, bioreactor, table to bioreactor.
It is a problem solving. For example, we need to cultivate the lactobacillus, convert and CFU. And then within the rest, then with or without the 24 hours, and for probiotic production. and define the success rate is sterilization and the many other also. And it is used is also to the reculture method and also the degradation method is 5%.
Ideate part. In this part, in this part we can produce the microbes, extremely environmental microbes at low cost. First we choose the culture media, anything such as the rich culture media, minimal culture media, waste culture media, etc. And choose the source of the carbon energy. Carbon energy helps to growth of the microbes. And example such as the lactose, cellulose, maltose, glucose, lignocellulose, etc. And the important one of the growth of the microbes is temperature. Microbes are growth at optimal temperature. The low microbes are living at 15 degrees Celsius. To high microbes are developed the 80 degrees Celsius. Because we consider that 15 to 80 degrees Celsius are the best temperature to growth of the microbes. pH level and oxygen levels. Example, right pH and temperatures goes to the conditions to produce the aerobic and anaerobic conditions to produce the oxygen level. And in the aerobic conditions, microbes also growth. And anaerobic conditions also many microbes are growth. So we can consider the pH and the pH level and the oxygen level is important. And next, finally, cultural format is important one of things. So we can use the sacred flask and the biopractice use of this procedures.
In prototype, mainly use cheap and copper source first at a low cost small scale test to measure whether the optical density at 600 nanometer, pH, colony forming units and metabolites. And also after 12 to 48 hours, observe whether it's include positive or negative, and also checks the sterility.
And the run experiment and character real data, and I have a best prototype in a triplicate and monitor through a dissolved oxygen and dissolved pH and biomass. Check possible to contamination and check the coefficient of pH.